Yeah, sperm has always been an enigma, and andrology as a science is developing rapidly, basically because we have the semen sample and we can dissect it and do a lot of tests, whatever we want, unlike the oocyte. So, consequently, the belief that the sperm is competent on the basis of external characteristics is something which is spurious, which has its own fallacies, and there has been a constant attempt to improve the basic test, which is a semen analysis, which I compare something like an ECG. So there has been a constant attempt to improve the diagnosis of treatment of the male fertility problems, and all of us nowadays realize that semen analysis is an extremely imperfect test. It's a mass screening tool. It's not an indicator or a barometer of fertility potential of an individual. And we do know that sperm DNA fragmentation has become one of the widely used complementary tests in order to assess the sperm internal integrity or the fertilizing capacity. And we also know that there are several studies which have demonstrated that the sperm genetic defects are associated with infertility, recurrent abortions, and whatnot. And in order to select a sperm, we only had a method of crude separation, which is the density gradient and a swim up, which are basically based on the capacity of a motile sperm penetrating a media which we normally use. And the controversy is that can we select, now that we know that DNA is an integral part of fertilization, can we select the sperm on the basis of DNA fragmentation? Unfortunately, DNA fragmentation is not a perfect test. This method detects late apoptosis of the sperm, but we need better understanding of all stages of apoptosis, which is far more informative. That's where the research is headed. And one of the earliest markers of apoptosis is the loss of membrane integrity, which leads to phospholipid phosphatidylserine externalization, a molecule which has a high affinity to annexin V. So consequently, annexin V based apoptosis detection and a couple of other methods are gaining prominence in order to identify a perfect sperm. Because if you have a perfect sperm, the belief was that sperm is not important, but I do believe sperm plays a major role. So many studies have evaluated the use of magnetic activated cell sorting with annexin V binding, which is a method to reduce or identify early apoptotic sperm and improve the sperm and the embryo quality. And there are several studies which and even meta-analysis which have suggested, regardless of the DNA fragmentation results, apoptotic sperm is the best way to go about it, and DNA fragmentation is not the only way to go about it, though given if DNA fragmentation gives you an idea of the chance of apoptosis. So, and if you look at the meta-analysis data of using magnetic activated cell sorting in ART, there has been a statistically significant difference in the pregnancy rate when you study the group which has used MACS and when you have the control group. But unfortunately, there is no difference in the implantation or miscarriage rates, which is what we normally would have expected it to happen. And this is a summary of several studies which were done. And as you can see, there is a significant difference in the fertilization or the pregnancy rate, but not in the implantation or the miscarriage rate. And the pregnancy rate seems to be far higher. Very few people have studied live birth pregnancy rate, and that is one of the fallacies of the various studies which have done. But definitely there is a role of using the sperm, which seems to be better after using magnetic activation with annexin V binding. I'll explain to you the procedure how we do it. And this is compared, there have been about almost 2,000 patients who have undergone in several studies, and it definitely shows a significant improvement in the sperm characteristics. So at this point in time, magnetic activated cell sorting is an efficient method that can avoid apoptotic sperm when you want to select a proper sperm. And magnetic activated cell sorting, or MACS, efficiently reduces sperm DNA fragmentation levels and also effectively separates apoptotic from non-apoptotic spermatozoa. And this definitely leads to an improvement in the sperm quality and functionality, because it positively affects the sperm motility and morphology as determined by your sperm deformity index. And there have been improvement in some authors of fertilization rates and embryo quality when they grade the embryo, because the best sperms were selected using this technique than with any other standard method, either density gradient or your swim up or whatever. However, there has been no difference in the fertilization rates. Another benefit of this technology, it may improve the ART results, is the efficient removal of caspases that are present in the human spermatozoa, which represent the main pathway for apoptosis, and this is detrimental to even the embryo. So this removal of caspase enhances the human sperm motility and cryosurvival rates following cryopreservation also. So there's a lot of role of using this kind of a sperm. The ideal sperm preparation method ideally should select the best sperm, and we all agree with that. And most studies demonstrate that the double density gradient centrifugation combined with MACS may be the most advantageous method of sperm selection. But unfortunately, MACS has its limitation, though it has been one of the most promising techniques which have been used in assisted reproductive techniques. Unfortunately, IUI has been excluded basically because MACS as a technique takes a long time to do. It takes about an hour for you to separate the sperm, and consequently it is predominantly used in selecting the sperm for an ICSI procedure, especially in men with high DNA fragmentation, and with the hope that MACS technology would improve the results for the couples in this kind of a scenario. But what is the evidence, and are we sure that MACS is here to stay? Though there was no significant discrepancy between the results of these studies, none of them considered the female factors, and these studies did demonstrate considerable variability. So the jury is still out there. The variability is one of the issue, and also the small sample size is the other issue. And of course, we don't have live birth rate data, and a great sample of patients is needed to determine the usefulness of this technique and whether it is clinically beneficial sperm selection method in assisted reproduction. Just to summarize, MACS is a technology which is exciting, and it's a cumbersome technology at this point in time, also on the expensive side. It takes about an hour for you to separate the sperm, so there is a fear that you may lose some of the motile sperm in the process. So it is still predominantly used only in an ICSI setting. And as you can see, this is the MACS separator where you have a magnet and a MS column and a magnet, and usually you tag 550 nanometer beads which are biodegradable, which is conjugated to annexin V, and you pass it through a strong magnet, and essentially the sperm gets labeled because of its affinity to phosphorylylene. So annexin binds to that, and consequently you can separate because of the trapping of the cells by the magnet, and this is a very high powerful magnet. So you can use the the cells of interest which is separated, which usually are non-apoptotic cell. Now the question is this the only technique? Are there other techniques of separating an early apoptosis? And this is where considerable research is going on. And the other technique which I want to discuss or tell you is the magnetic bead-based separation of sperm from semen sample based on Halyardon binding capacity. Gabor Huzar some time ago designed a dish called Pixie, and which is used, it was basically used for it is a Halyardon slide, and it is used for selecting the best sperm for an ICSI. Now, we know that Halyaronan is a major component of the extracellular matrix of the cumulus, this is one of the principal components, and the Halyaronan forms a complex with glycodylin, which is an interacting protein which retains and concentrates glycodin C, which is essential for sperm zona binding. So HA receptor is incorporated in the sperm during the last stages of maturation. And that is why it is critical, because that means you don't have apoptosis in any of the stages, and probably it is the most mature sperm. So HA binding has been used for decades now, but the process of HA binding is evolving, is an indication of sperm maturity and exhibit enhanced chromosomal and DNA integrity. And HA bound sperms, like I said, it was almost 5 years ago when Gabor Huzar designed the dish called Pixie, but has never been used for IUI. Or for selecting a sperm of a motile sperm concentration when you want to do simple procedures. The reason for that is extracting on the basis of binding to a slide. That means you are restricting the number of slides, number of sperms which you can select after binding in a pixie dish. So what we have done, or we are trying to do, where we have patented this technology, is Microfluidics platform to extract and the mature and motile HA binding sperm from semen samples on a microfluidic platform, and to automate this process and get a simplified method of existing from an existing complex protocol. And this process hardly takes 5 minutes to do, and it is operator independent. So what we use in this is paramagnetic beads which are functionalized with biotin streptavidin complex. This is used to immobilize the HA molecule, and this complex is incubated with sperms, and the bead HA sperm complex is separated by using a magnetic field. And I'm sure there is a lecture by Dhananjay Dandukuri with whom I collaborate, and he will be talking about the microfluidic principle later. And this is how now suspended in Halyardon sperm, and that's how it is isolated. And what we use is immiscible filtration assisted by surface tension. That is the technology which uses the surface tension principles, and we use 1 or 2 simple washes, and then you have sperm which are HA positive sperm. So this is a simple way of this is a microfluidic platform where you pass the sperm in one stream, and you pass the HA magnetic beads, which are Halyardon beads, in another stream, and both of them interact at one point, and those sperms which are HA positive will get bound, and again they get separated into a third stream, and consequently you will get 2 streams. One is HA positive sperm, the other is HA negative sperm. Like I said, this is operator independent, and it is a simple process, and it's not complex, and it doesn't take much time. So it's ideal for all sorts of sperm separation techniques. So just to conclude, sperm preparation based on HA binding is probably the most exciting technology, and is possible by using simple techniques, and this can be automated and hence made technician independent. And this is one of the challenges in andrology, basically because you require a lot of quality control. Interoperator variability is more than 15% in andrology labs, and that is unacceptable as a biochemical test anywhere in the world. And time taken to perform this in the lab will be less than 1 hour against the standard conventional technique which you take for 1.5 hours. This is including the preparation and all these processes. So the new technologies, the take home is there are several new technologies on the horizon which will offer probably robust and non-apoptotic sperm selection, which is the need of the day. This will eliminate interoperator variability by offering consistent results, and that is also the need of the day, and this will reduce consequently manpower, supervision and training. Thank you very much.